Hello guys, welcome back to my YouTube channel. From this video, I will discuss about the neurulation process. Before going into neurulation process, I will summarize the previous video about the gastrulation. So in this diagram, you can see the blastocyst here. First diagram, you can see the blastocyst with the two layers, epiblast and the hypoblast. You can see the bluish color one, always the epiblast, and yellowish color one demonstrate the hypoblast. And here is the cross section, and then you can see the primitive streak formation, and the primitive node here. From the primitive streak, cells begin to invaginate. Cells in the epiblast begins to invaginate downwards or the inwards and replace hypoblast cells, forming endoderm. In between the epiblast and the hypoblast, it forms the mesoderm, and replacing the epiblast cells, it forms the ectoderm, the three germ layers, ectoderm, mesoderm, and the endoderm from gastrulation process. Now we will see the neurulation process, that means the neural tube formation. Hope this diagram is clear for you. Here you can see the yolk sac, then amniotic cavity, and the three layers, ectoderm, mesoderm, and the endoderm. Here the lower part you have the coronal weave, and the upper part you have the superior weave. So by the end of the third week, lateral edges of the neural plate become elevated to form neural folds. Here is the ectoderm. In the ectoderm you have the neural plate, and the lateral edges of neural plate become elevated to form neural folds. Here from the coronal weave you can easily demonstrate, you can easily see the formation of neural folds. The lateral edges of the neural plate becomes elevated and forms neural folds. Here it forms neural folds. Here is the neural plate. Here is the neural plate in the ectoderm. Here is the primitive streak, primitive node, then notochord and notochordal knot. Here is the cranial end and the caudal end is there. Then this is the neural plate. The lateral edge of the neural plate is elevated to form neural folds like here. Then depressed mid-region forms neural groove. Here is the depressed area, and this area is called as the neural groove. And then gradually neural folds approach each other in the midline and fuse together. Here you can see the elevated edges, and here you can see they fuse in the midline. Here you can see the after fusion of the in the midline. Here like this comes in the midline and this comes in the midline. They meet together and fuse like here. So what happens in neurulation? There is a neural plate in the ectoderm, and the lateral edges become elevated to form neural folds, and the depressed mid-region is called as the neural groove, and the two lateral edges or the neural folds approach together in the midline and they fuse together, forming the neural tube. This fusion begins in the cervical region and proceeds cranially and caudally. Initially, you can see this fusion begins in the cervical region. Here, this fusion begins in the cervical region and it reaches cranially and caudally gradually. As a result of fusion of cranial direction and caudal direction, it forms the neural tube. Until fusion is complete, cephalic and caudal ends of the neural tube communicate with the amniotic cavity by the way the anterior or the cranial and posterior or the caudal neuropores. These are the neuropores. Here, these are gradually fusing. These are gradually fusing like in cranial direction and caudal direction. Here you can see the most of areas has been fused and small area is remaining. This remaining area is called as the anterior or the cranial neuropore. In the caudal direction you have the posterior or the caudal neuropore, which communicates with the amniotic cavity, because still they have not closed together or fused together. Actually, the closure of cranial neuropore occurs at approximately day 25, whereas the posterior neuropore closes at day 28. About here you can see the about day 24 to 25 anterior or cranial neuropore fuses together, and posterior the caudal neuropore closes at the day 28, 26 to 28. Neurulation is then complete and the central nervous system is represented by a closed tubular structure with a narrow caudal portion, the spinal cord. And the much broader cephalic portion is characterized by number of dilatations, which are called as brain vesicles. Okay, now you know the formation of neural tube. Um, During the formation of neural tube, you have to know the formation of neural crest cells as well a little bit. As the neural folds elevate and fuse, the cells at the lateral border or the crest of the neuroectoderm begins to dissociate from their neighbors. You can see here they begins to dissociate in this area, begins to dissociate like this, and it forms neural crest cells. So that's all about neurulation process. In summary, what happens is there is the ectoderm, and in the ectoderm you have the neural plate. Here the lateral edges of the neural plate elevated to form neural folds, and gradually they elevate and fuse together in the midline forming neural tube. In the midline groove you have the neural groove also, and during formation of neural tube there are neural crest cells are formed. First, the fusion of the neural tube starts from the cervical region and it proceeds in cranial direction and caudal direction. And during this proceeding, there is remaining part in the anteriorly cranial neuropore is there and posteriorly caudal neuropore is there. The cranial neuropore closes at about day 24 to 25, and posterior the caudal neuropore closes at the day 26 to 28. This is the process of neurulation, and thank you for watching.